All right, we're going to try this a little different. So for our lecture on DNA history, I'm going to post this for you on Canvas. So let's do just a brief overview. We're going to watch a video in class about it. We're going to get, and then we're going to move actually into what really matters, which is how DNA is replicated and the structure and everything like that. So let's just talk about the history component now, because it's important to introduce you to some interesting people, some people that have made what we know today out there. So again, just like always, what our big questions are for this is, what does DNA look like? How does it work? And what is the structure and function of DNA? And how is it determined? And so these are the people that got us there. So back in the 20th century, mid-20th century, there was the real discovery, the idea that there was something that was making the inheritance that we've talked about before kind of come to light. And so we don't really know what it was. No one quite understood how this process worked. We understood the probability because of Mendel, but there was no real understanding of what it was that we were looking at the probability for. So first person to really look at that was Friedrich Miescher, who is the person who discovered what nucleic acids were back in the 1800s. And that led us then to the understanding of chromosomes, that these nucleic acids were building blocks for long chains of nucleic acids, which ultimately led us to the understanding of chromosomes. And chromosomes, of course, are DNA wrapped around histone proteins. And so it is kind of an amalgamation. And remember, we call that chromatin as it sits inside the nucleus. But we didn't really understand the structure or the function of DNA. We just understood that there was this formation of it, and this had something to do with it. So the first person to really start working with it was Frederick Griffith, who did what's called transformation, and through transformation was able to discover that there were heritable traits through this DNA. And what he did is he could give information from one bacteria into another bacteria. We could actually turn it into what we wanted. However, that was kind of where he left it. We figured out that we could do it, but he didn't do a whole lot more with it after that point. And so he used strep, actually was the bacteria that he utilized for this. And he infused it into mice, which strep, as you all know, many of you have had it, leads to an infection that in some cases, if left untreated, could actually kill you. So he did it, he injected mice with it, and he heat killed certain cells within the bacteria and found that he could actually turn those healthy cells into bad cells by transforming the bacteria. So that was the experiment that he did, Griffith. And so then, I'm going to go back here. So after Griffiths did that, we have three other scientists who came along who took Griffith's experiment and refined it, and that was Avery, McCarthy, and MacLeod. And so you see the photographs of the three gentlemen here. They took Griffith's experiment and started to actually work with it and move it one step further. And ideally, what they were really trying to determine is if it was protein-based or if it was nucleic acid-based that was being moved from one bacteria to the other. So they exposed the strains of bacteria that Griffith had to pure strains with proteins to see if the proteins were moving or if the nucleic acids were moving. And so then later, that led to Hershey and Chase to do what's called the blender experiment and to actually really get even further into determining if this DNA, which at the time didn't really have a term, it was just heritable traits or chromosomes, if it was able to be the component that we expected it to be. And basically what they did, is they worked with bacteriophages. And I believe we've talked about phages before, but phages are viruses that infect bacteria. And viruses are strands of DNA or RNA that you can use to infuse into bacteria, and it changes the bacteria. And so they use these bacteriophages to sort of determine, and what they did is they tagged DNA and proteins with phosphorescence. And so what they did is they tagged specific components of the DNA and specific components of the proteins. And we've talked about this before. One has one thing, one has the other. Proteins, remember, have sulfur components, and DNA has phosphate components. So what they did is they tagged phosphates for one reason, and then they tagged sulfur for the other. And whatever was being transferred from one species to the other and being infused into the DNA was going to tell us if it was proteins or if it was nucleic acids that were actually moving on. Because if the bacteria, what they did is they took the viruses and they phosphoresced sulfur in one and they phosphate in the other, and they inject it into the bacteria. And whichever one phosphoresced in the bacteria, whether it be the sulfur or the bacteria, the sulfur or the phosphate, that's what told them what it was. And of course, sulfur didn't phosphoresce in the bacteria because the protein itself was not being transferred on, but the phosphates were. And so that's how we came to realize that it was the nucleic acids that were actually being passed on from one generation to the next. Once that became known, we understood what those nucleic acids were, which then we found out, of course, A, T, C, G, down the line. Then there was work that was starting to get done on the ATCG. And so you had Erwin Chargaff, who was one that was looking at the ACTG and trying to figure out the ratios of these and how those ratios compared to things. So Chargaff, who was an Austrian biochemist, he looked at the rules of DNA composition. He looked at how many A's there were against how many T's and so on, and he's the one who realized that A bonds with T and C bonds with G, and he noticed it in a percentage value. So what he found was that if you take a look here at this chart, that if we have E. coli, which was our species here, 24.7%, or about 25% of the DNA was adenine, 26% was guanine, 25% was cytosine, 23% was thymine. And what he basically found is that the percentage of thymine and adenine was much closer, and the guanine and cytosine was much closer. So the idea that they were the ones that were partnering up. And so we have what's called Chargaff's ratios. And so we can actually determine the amount of everything, even if we just have one number. Let's say we just have adenine, for example. We know that there's 24% of the DNA is adenine. Then what we do is we say, okay, well, 24% is adenine. That means 24% is thymine, which means 48% are those. That means that the alternative is 52% is guanine and cytosine, and we divide that in two, which then tells us how much guanine and cytosine is present. So this becomes what's called Chargaff's rule. So we're able to actually mathematically calculate the values out. Watson and Crick, of course, are ones that you know in 1953, and then Franklin and Wilkins are the other two. Franklin, hopefully you know by now. And these were the ones that actually started to determine what this DNA was structured like. So we know that it had all these nucleic acids, but how did it look? And so Franklin did the first X-ray. This is a photograph of the very first DNA X-ray, where we get the idea of the double helix. And we actually start to see the double helix, and we start to determine what the double helix is and how that whole process works. So Watson took that X-ray photograph, she shared it. Franklin took the photograph, she shared it with Watson and Crick, and they were the ones who actually came around and published the double helix, and later on received the Nobel Prize for that. So here's the model of the double helix as we know it today. So the puzzle of DNA was solved. We understand how it was structured. It was made out of those nucleic acids, how those nucleic acids came together, and then ultimately how they wound around each other to create the double helix. So the two key components here, and we're going to get into more details of the structure later, is that we understand that the phosphate groups and the sugar groups kind of back against each other, making the backbone. And the two pieces that are the most important are the nucleic acid component, which is this, and how they hydrogen bond with each other. And so we're going to talk a little more about that structure. We'll do a little activity later on about that structure and how that structure works. But this gives you sort of the basic understanding. Now, looking at these components, and we look closer at these, when we look at the phosphate and the sugar molecule, and how this sugar molecule bonds to this phosphate, and this one, and this one, and this one to make that alternation, we call those phosphodiester bonds. We've talked in the past about glycosidic linkages and peptide bonds and things like that. So in nucleic acids, the bond between the phosphate and the sugar is what we call a phosphodiester bond. So you see the word phosphate in there, and diester is a term for sugar. So the phosphodiester bonds are covalently bonded back and forth in alternating pairs. The opposite strands, the base pair, becomes the base pair, the adenine to thymine, guanine to cytosine. And we see those here. Notice, and if I can get a little closer for you, if you notice, the adenine is double ringed, cytosine and thymine are single ringed, and guanine, of course, is double ringed. And so that's important later on when we talk about the structure of of DNA. To be able to do is to be able to explain how the structure of DNA is related to its function, explain the contributions of those scientists that into our understanding of DNA and the historical developments that led to that scientific discovery. And it's important that you know that because those are names that will appear at some point again, and so you want to make sure you know them, just like we knew Mendel, Darwin, and some of the others that we've talked about up to now. So I hope that helps. Thanks so much.